What if one of biology's most stubborn mysteries, something that absorbed nearly 80 years of experiments, arguments, failed models, and slow, careful progress, could now be approached with a single line of text? Today, that's not an exaggeration. A researcher can sit down at a laptop, copy a protein sequence from a database, a long, unbroken string of letters representing amino acids, paste it into a web interface, press enter, and within seconds watch a detailed three-dimensional structure appear on the screen. A problem that once demanded years of specialized laboratory work can now, in many cases, be explored in the time it takes for a progress bar to fill. 80 years of human effort, one prompt. That contrast is dramatic, but it only makes sense if we understand what those 80 years were spent trying to solve. Proteins are the molecular machines that make life possible. Every cell in your body is filled with them. Some act as enzymes, speeding up chemical reactions that would otherwise happen too slowly to sustain life. Some serve as structural components, giving cells their shape and stability. Others act as messengers, receptors, transporters, or motors. Hemoglobin carries oxygen through your bloodstream. Antibodies recognize invading pathogens. Ion channels regulate electrical signals in your neurons. In every case, function depends on structure. A protein begins as a linear chain of amino acids, assembled by ribosomes, according to the instructions encoded in DNA. This chain is chemically simple in one sense. It's just a sequence built from about 20 different kinds of amino acids. But once synthesized, it doesn't remain a loose string. It rapidly folds into a specific three-dimensional shape. Parts of the chain coil into spirals known as alpha helices. Other segments align into flat sheets. Distant regions of the sequence may come into close contact, forming intricate folds stabilized by a web of chemical interactions. This final 3D structure determines what the protein can do. It creates pockets where small molecules can bind. It positions chemical groups precisely so reactions can occur. It forms surfaces that interact with other proteins. If the structure changes, even slightly, the function can change or fail. When proteins misfold, the results can be devastating. In Alzheimer's disease, certain proteins aggregate into plaques after folding improperly. In cystic fibrosis, a mutation in a single protein disrupts its folding and prevents it from reaching the cell surface where it is needed. Many cancers involve proteins whose structures enable abnormal signaling or unchecked cell growth. The difference between health and disease can hinge on the precise arrangement of atoms within a protein. For much of modern medicine, knowing a protein's structure has been a gateway to understanding and intervention. Drug discovery often begins by studying the shape of a target protein in detail. If you can see the grooves and cavities on its surface, you can design molecules that fit into those spaces and alter the protein's behavior. Without structural information, drug design becomes guesswork. And yet, for decades, the only reliable way to obtain that structural information was through demanding experimental techniques. In the mid-20th century, X-ray crystallography became the dominant method for determining protein structures. The process required extraordinary patience. Scientists first had to purify the protein of interest, often from complex biological mixtures. Then they needed to persuade that protein to form a crystal, a highly ordered lattice in which millions of identical molecules line up in the same orientation. Many proteins refuse to crystallize, making this step a major obstacle. If crystals were obtained, researchers would expose them to X-rays and measure the diffraction patterns produced as the rays scattered off the electrons in the atoms. These patterns appear as arrays of spots on a detector. Beautiful, but cryptic. From those spots, scientists had to reconstruct the three-dimensional electron density of the protein and then build an atomic model that fit the data. It was part physics, part mathematics, part informed intuition. Solving a single protein structure could take years. It required specialized equipment, expertise, and persistence. Entire careers were built around determining a handful of structures. Later, methods such as nuclear magnetic resonance spectroscopy and cryo-electron microscopy expanded the toolkit, but they were no less sophisticated or resource-intensive. Cryo-EM, for example, involves flash freezing samples and imaging them with powerful electron beams, then reconstructing 3D structures from thousands or millions of noisy particle images. Over roughly 80 years, structural biology advanced step by step. The Protein Databank, a public repository of solved structures, grew steadily. Each new entry represented months or years of effort. Meanwhile, a fundamental question loomed over the field. If the amino acid sequence contains the instructions for folding, why can't we compute the structure directly? This became known as the protein folding problem. The premise is straightforward. Given a sequence of amino acids, can you predict the exact three-dimensional structure the protein will adopt under physiological conditions? Early experimental evidence suggested that the sequence should be enough. Classic refolding experiments demonstrated that some proteins, when unfolded and then returned to favorable conditions, spontaneously refold into their original shapes. That implied the necessary information was encoded in the linear sequence itself. No external template was required. In theory, the answer lies in physics. Each amino acid has chemical properties. Some are hydrophobic and tend to avoid water. Others are charged and interact with oppositely charged partners. Hydrogen bonds form between certain groups. Atoms repel each other at very close distances and attract each other at intermediate ranges. All these forces combine to create an energy landscape. The protein's folded structure generally corresponds to a low-energy state, where the arrangement of atoms is stable. So why not calculate it? The obstacle is combinatorial complexity. A protein with just 100 amino acids has an astronomical number of possible conformations. Each bond along the backbone can rotate within certain limits, and each rotation multiplies the number of potential shapes. Even if you drastically simplify the possibilities, the total number of configurations quickly becomes unimaginably large. A famous thought experiment illustrated the challenge. If a protein were to sample every possible conformation randomly in search of the lowest energy state, it would take longer than the age of the universe to find the correct one. Yet in living cells, proteins fold spontaneously in milliseconds to seconds. They do not explore all possibilities. They follow constrained pathways through the energy landscape, guided by local interactions and thermodynamic principles. Capturing that process computationally proved extraordinarily difficult. Over the decades, researchers developed increasingly sophisticated models. They created energy functions to score how favorable a given structure might be. They used molecular dynamics simulations to model atomic motions over time, calculating forces at each step. They incorporated knowledge of recurring structural motifs, recognizing that certain patterns, like alpha helices, appear frequently and can often be predicted from sequence. Another strategy involved comparative modeling. If a newly discovered protein had a sequence similar to one with a known structure, researchers could use the known structure as a template and adjust it accordingly. This worked well when close relatives existed, but it struggled when proteins had no obvious structural cousins. Computing power improved dramatically over the years, enabling longer and more detailed simulations. Yet despite these advances, predictions for many proteins remained imperfect. Some were reasonably accurate, others deviated in critical ways. The most challenging targets, proteins with novel folds or limited evolutionary relatives, remained elusive. To measure progress objectively, the scientific community created the CASP competition in the 1990s. CASP stands for Critical Assessment of Protein Structure Prediction. It was designed as a blind test. Organizers would collect sequences for proteins whose structures had been solved experimentally but not yet released publicly. Research groups would submit predictions based only on the sequences. After submissions closed, the experimental structures would be revealed, and predictions would be scored. CASP became a benchmark for the field. Every 2 years, teams gathered results and compared methods. Over time, scores improved, reflecting gradual refinement of algorithms and better use of data. But for decades, the improvement curve was steady rather than explosive. For the hardest targets, there was still a noticeable gap between prediction and experiment. Then, artificial intelligence entered the picture in a new way. Deep learning had already reshaped fields like computer vision and natural language processing. Instead of encoding explicit rules for recognizing objects or translating sentences, researchers trained neural networks on massive datasets. These networks learned complex statistical patterns directly from examples. With enough data and computing power, they often outperformed traditional rule-based systems. Structural biology had accumulated exactly the kind of dataset that deep learning systems could exploit. The protein data bank contained tens of thousands of experimentally determined structures. Sequence databases contained millions of related proteins across species. Evolutionary relationships among these sequences held subtle clues. If two amino acids in different positions tend to mutate together across many organisms, it often suggests they interact physically in the folded structure. The insight was that a neural network might learn to interpret these correlations and infer spatial relationships directly. AlphaFold, developed by DeepMind, embodied this approach. It combined information from multiple sequence alignments, capturing evolutionary patterns, with neural network architectures designed to reason about pairwise relationships and geometric constraints. Rather than simulating the folding process step by step, it predicted distances and angles between amino acids and iteratively refined a 3D model consistent with those predictions. When AlphaFold participated in CASP, the results were striking. For many proteins, especially some of the most difficult ones, the predicted structures were dramatically closer to experimental results than those from previous methods. In numerous cases, the accuracy approached that of experimental techniques themselves, at least for the core regions of the proteins. For a field accustomed to incremental progress, the jump felt transformative. 80 years of cumulative experimentation and algorithm development had laid the groundwork. Now, with one prompt, a trained model could deliver a structural prediction that might once have required years of laboratory work. The impact extended beyond the competition. As predicted structures were released publicly for vast numbers of proteins, researchers gained access to an unprecedented structural atlas. Instead of focusing on a handful of well-studied proteins, scientists could explore entire proteomes, the complete set of proteins expressed by an organism. In drug discovery, this meant faster hypothesis generation and target analysis. In basic biology, it meant structural insight into proteins that had never been experimentally characterized. In fields like microbiology and environmental science, it opened doors to studying enzymes from obscure organisms that might hold practical applications. At the same time, it's important to recognize the foundation beneath this shift. The neural networks did not invent protein physics from scratch. They learned from decades of experimental structures generated by thousands of scientists worldwide. Each crystal grown, each diffraction pattern analyzed, each cryo-EM reconstruction contributed data that made large-scale learning possible. The one-prompt capability is built on that mountain of work. And the story is not over. Predicted structures are immensely useful, but they represent models, often of proteins in isolation and in a single conformation. In living cells, proteins are dynamic. They interact with other molecules, change shape upon binding, assemble into complexes, and operate in crowded environments. Experiments remain essential for probing these dynamics and for validating unexpected or high-stakes predictions. Still, the contrast remains powerful. A problem that defined structural biology for nearly a century can now, in many cases, be addressed by entering a sequence into a computational tool. 80 years of patient, meticulous science. One prompt on a screen. It's not a story about machines replacing scientists. It's a story about accumulation, about how decades of careful human effort can be distilled into systems that amplify our reach. The scientists who solved structures by hand built the dataset. The researchers who designed algorithms refined the questions. The engineers who developed neural networks created tools capable of absorbing patterns at a scale no individual could manage. Together, they transformed a generational scientific struggle into something that can be explored in seconds. And somewhere right now, a student is rotating a predicted protein structure on a laptop, zooming in on an active site, forming new ideas. They are benefiting from 80 years of collective effort, condensed into a single prompt, and a model that learned from everything that came before.